This is the super fast-paced review video for D3.1 and D3.2. Let's begin with D3.1 Reproduction. First up, let's contrast asexual and sexual reproduction. Asexual reproduction happens when there's one parent. It occurs through mitosis, where one parent becomes two genetically identical offspring that are also identical to that parent. Okay, existing gene combinations are maintained. There's no genetic variation. Benefit, well, if that organism is already adapted to an unchanging environment, the offspring are also adapted. On the other hand, sexual reproduction happens when there are two parents. It occurs through meiosis, okay, where there is division of that parent's chromosomes in half. Okay, so that we end up with then a joining of two halves of chromosomes from one from each parent, okay, into one whole offspring, which is now genetically different, okay, from other offspring as well as from their parents. New gene combinations are produced. We now have genetic variation. And the benefit from this is if those offspring are in a changing environment, those offspring may be better adapted than their parents. Okay, the fusion of a male and female gamete is called fertilization. Okay, it occurs the instant that a new individual is formed. Fertilization doubles the number of chromosomes because we're joining two different gametes, and so meiosis has to occur first, or the chromosome number can be halved so that two halved gametes can become one whole zygote. Okay, let's look at the difference between male and female gametes. Male gametes travel to the female. They're smaller. Okay, they have less food reserves, only enough for the gamete. Okay, the reproductive strategy is to create large quantities of sperm with minimal investment in each sperm. Okay, in contrast, we have a major distinguish question we could have here. Female gametes are sessile. They are not moving towards the males. Okay, they are much larger in size. Okay, they have more food reserves, enough for development of an embryo. And the reproductive strategy for females is going to invest in fewer, more energetically costly eggs to ensure offspring survival, okay, with the priority of selecting high quality of males. Okay, now we do have two drawing skills here, the human male and female reproductive systems. Now, I don't think it's likely that IB would actually ask you to draw these, but they do have them listed as a drawing skill, not just the actual drawing, but also having the functions annotated. I don't think they're going to ask you to draw it, but knowing how to draw it and knowing the functions is going to allow you to answer a lot of different potential questions that they could ask you. Okay, so there's the drawing skill right there. Let's take a look at some of the functions. Okay, so you should pause your screen now, read through all of these different structures, make sure you can identify the function of each. Now the same holds true for the female reproductive system. Okay, technically it's a drawing skill with being able to annotate the function. Okay, this is going to help you to be able to answer a lot of different questions that could potentially be asked. Okay, so here then again are all of those structures and functions. You should pause here and make sure that you know all of them. All right, fertilization in humans. This is when sperm meets egg and then forms a zygote. Okay, the plasma membranes of sperm have receptors that detect chemicals released by the egg, and so that enables the sperm to directionally swim towards the egg. Now we're going to see today that the egg is surrounded by a cloud of follicle cells here in orange. Okay, there is also a layer of glycoproteins called the zona pellucida here in this purpley blue color. The sperm has to push its way between that cloud of follicle cells and then make its way through the zona pellucida in order for fertilization to occur. Once it gets through the zona pellucida by digesting those glycoproteins that made it up, okay, using enzymes that are in this acrosome, which we'll see again in the future, okay, we're going to see that it's able to bind to the plasma membrane of the egg cell, fuse together, and this is the moment of fertilization. Now, immediately after fertilization, that layer of glycoprotein hardens to prevent entry of more sperm. if more than one sperm fertilizes an egg, it becomes unviable. Okay, we need to make sure that only a single sperm penetrates that egg. And so there are two major reactions that occur during fertilization, and we're going to look at those a little bit further down the line. Okay, now the sperm tail either doesn't penetrate the egg or gets broken down inside the zygote. Same goes with the mitochondria. Okay, and so all of the organelles that are in the egg are coming, or all the organelles in the eventual zygote are coming from the egg. Okay, so let's now look at the menstrual cycle. The menstrual cycle is commonly a 7 or 8 mark question, and so it's a good idea to know all of the hormones involved really well, like the back of your hand, okay, as well as how they provide feedback, both negative and positive feedback on other hormones. Okay, so the menstrual cycle occurs in women from puberty till menopause, stops during pregnancies or ill health. Okay, we're going to look at the uterine cycle and the ovarian cycle. Each time the menstrual cycle occurs, it provides the chance of a pregnancy, and this whole cycle is regulated by hormones. So let's begin by looking at the two halves of this cycle. Now, the first half we're going to call the follicular phase. This is because we have growth of follicles in the ovary. Okay, inside of each follicle, there's an egg that is stimulated to grow. Okay, and halfway through, okay, on approximately day 14 of the menstrual cycle, ovulation occurs and the egg is released. Now, the second half of the cycle is called the luteal phase. Okay, and this is where after ovulation, the follicle becomes what's called the corpus luteum, or the yellow body. Okay, it's going to be producing a lot of hormones that we're going to see later, especially if fertilization does occur. But if fertilization doesn't occur, the corpus luteum breaks down and the cycle begins again. The ovary returns to the follicular phase. Okay, let's contrast that then with what happens in the uterus. Okay, so as we can see here then down for D, okay, during the follicular phase, now the beginning of the menstrual cycle is menstruation, and that is because it's an easily recognizable stage. So day 1 is the beginning of the period where there's the shedding of the endometrium. The endometrium is the uterine lining. Okay, this lasts anywhere from 4 to 7 days before the lining of the uterus begins to thicken again. Okay, and then it stays thickened during the luteal phase to prepare for any potential fertilized egg to be implanted, because remember, ovulation happened right in the middle at day 14, and so for these next few days, those endometrium is ready for the implantation of a fertilized egg. If that doesn't occur, however, okay, it breaks down, it's shed during menstruation, and on and on and on we go. Okay, now let's talk about the 4 hormones that drive these events in the ovary and in the uterus. Okay, first up, we're going to learn about follicle stimulating hormone, or FSH, and luteinizing hormone. They are both produced in the pituitary gland at the base of the brain. They bind to FSH and LH receptors in the membranes of the follicle cells. Okay, then we have our ovarian hormones. Okay, estradiol and progesterone. Now, in the old curriculum, we always referred to this as estrogen. Estradiol is a form of estrogen, and this is how it's referred to in the guide. Okay, however, I noticed that even in the specimen paper, they're still referring to it as estrogen. So I think you're pretty safe to refer to it either way, but because the guide does refer to it as estradiol, I would say if you're only going to choose one or the other, go with estradiol. Okay, so these are both our ovarian steroid hormones. They're produced by the wall of the follicle and then the corpus luteum that develops from it. Okay, they're going to influence gene expression and therefore development in the uterus and many other parts of the female body. So let's look at these 4 hormones in a little bit more detail. Let's start with those 2 pituitary hormones. Okay, FSH, its primary job is in its name. Follicle stimulating hormone stimulates the development of follicles. So that one should be easy to remember. Okay, FSH also stimulates secretion of estradiol by the follicle wall. Luteinizing hormone, as you can see here in red, comes to a sudden sharp peak towards the end of the follicular phase, okay, which is, remember, right when this ping ovulation occurs. And sure enough, that is luteinizing hormone's primary job, is to stimulate ovulation. Okay, LH also promotes post-ovulation development of the wall of the follicle into the corpus luteum, okay, which then secretes estradiol, which is a form of positive feedback, and progesterone. Now, let's look at those two hormones in a little bit more detail. Now, these are going to be our ovarian hormones. Okay, estradiol rises to a peak during the end of the follicular phase, as seen here. Its primary job is that it stimulates the thickening of the endometrium, that uterine lining. Okay, you can see here that as estrogen rises, so is the thickness of the endometrium. When it reaches high levels, however, okay, estradiol inhibits the secretion of FSH and stimulates LH secretion. So at high levels, right, we want LH to occur, we want ovulation to occur, and we don't want new follicles to develop because we're about to have ovulation occur and we're hoping that there might be some fertilization during the second half of the cycle. Now, progesterone levels rise at the start of the luteal phase, as seen here in purple. Okay, they reach a peak and then drop back to a low level at the end of the luteal phase. Progesterone's primary job is that it promotes the thickening and maintenance of the endometrium. So it maintains that thick endometrial layer, waiting for a potential fertilized egg to appear and embed in that endometrium. Okay, progesterone also inhibits FSH and LH secretion by the pituitary gland in negative feedback because, again, we don't want new follicles to be stimulated or new ovulation to occur while we're in this luteal phase, while we're waiting for that potential implanted egg. All right, now here is a previous question. I recommend pausing the video, read through. This is an old 8-mark question, but it goes through all of those different hormones and how they regulate the menstrual cycle. Now, the natural method of fertilization in humans is in vivo, meaning it occurs inside the living tissues of the body, vivo. However, fertilization can also happen outside the body in carefully controlled laboratory conditions called in vitro fertilization, technically in glass, usually abbreviated to IVF. Okay, this procedure has been used extensively to overcome fertility problems in either the male or female present. So let's run through the use of hormones in IVF treatments. Now, there are several protocols for IVF, but the first stage is usually downregulation. Every day for about 2 weeks, the woman has either an injection or a nasal spray containing a drug to stop the pituitary gland secreting FSH or LH. Therefore, secretion of estradiol and progesterone stops. This whole thing suspends the woman's normal menstrual cycle, which is then going to allow for doctors to control the timing and amount of egg production. Okay, so once downregulation has occurred, okay, intramuscular injections of FSH are then given daily for 7 to 12 days, stimulating follicles to develop. The aim is to generate a much higher FSH concentration than during the normal menstrual cycle to induce superovulation, where far more follicles than usual develop. Between 8 and 15 follicles is ideal, each containing an egg. Okay, when they reach a certain size, they're stimulated to mature by an injection of HCG, human chorionic gonadotropin. That hormone is normally secreted by a human embryo when it's about a week old as a signal to its mother it's alive and in need of sustenance from the endometrium. We're going to see it again later, okay, as the hormone that is used in pregnancy tests to determine whether a woman is pregnant. Now, in IVF, egg collection then is performed 34 to 35 hours after that HCG injection. This is a minor surgical procedure, takes about 20 minutes. A micropipette mounted on an ultrasound scanner is passed through the uterine wall to draw eggs out of the follicles. Each egg then in the laboratory is mixed with sperm cells in sterile conditions, incubated for a day, and if fertilization is successful, 1 or more embryos are placed in the uterus when they're about 48 hours old. Okay, because the woman has not gone through a normal menstrual cycle, extra progesterone is usually given as a tablet placed in the vagina to ensure that the uterine lining is maintained. Okay, and then if the embryo implants continues to grow, the pregnancy that follows is no different from a pregnancy that began by natural conception. Let's pivot now to looking at plants. Now, flowers are the part of flowering plants that are used for sexual reproduction. Meiosis, gamete production, fertilization all happen inside flowers. In mammals, the zygote produced by fertilization is retained by the female parent. It's supplied with food as it grows and develops into an embryo. In flowering plants, however, while the embryo develops inside a seed, inside of the female parent, it then gets dispersed from the female parent in hopes of not competing with the parent, and that seed can then germinate and grow in its new location. Okay, now let's outline both the male and female parts of the flower. Let's start with the male parts called the stamens. They're made up of an anther and a filament at the supporting stalk that holds the anther. Okay, now in the anther we have some diploid cells. They're going to divide by meiosis to produce 4 haploid cells, each of which develops into a pollen grain. Okay, those male gametes inside those pollen, have this thickened wall, and they are going to be transferred in many different ways, which we'll mention, to land on the stigma. Now, the stigma is part of the carpels, which are the female parts that consist of the stigma, style, and ovary. Okay, so again, the stigma is where that pollen is going to land. Okay, that pollen will then travel through a pollen tube down the style until it reaches the ovary where it finds the ovules. Okay, the ovary could have 1 or more ovules. They're multicellular. Okay, 1 cell in the center of the ovule grows particularly large, divides by meiosis to produce 4 haploid nuclei. Okay, and one of those, is going to be what becomes the female gamete or egg. Okay, can you pause the video and be able to identify all of these different parts of a flower? Next up. Successful sexual reproduction in flowering plants depends on pollination, fertilization, and embryo development. So first up, pollination is just the transfer of pollen from an anther, to a stigma. It can be moved by wind or animals. On the other hand, then fertilization happens inside an ovule in the ovary. So from each pollen grain on the stigma, a tube, as you can see here, will grow down the style to the ovary. The pollen tube carries male gametes near its growing tip. Once it's inside the ovary, the pollen tube grows towards an ovule and into one end of it. When the pollen tube reaches the center of the ovule, the male gametes are released and fertilization occurs. And so then lastly, we have embryo development. Once fertilization occurs, we have a zygote, a new diploid cell, which then develops into an embryo with an embryo root, an embryo shoot, and either 1 or 2 embryo leaves called cotyledons, from where we get the term monocot and dicot. The type of reproduction that happens inside flowers is sexual because there's meiosis and gamete production followed by fertilization. Now, most flowers are hermaphrodite, meaning they have both male and female parts, so they can act as both a male parent and a female parent. Less commonly, some flowers have either male or female parts rather than both. The male and female flowers could be on the same plant or on separate plants. But either way, reproduction using flowers is sexual. So pollination usually occurs by insects, but can also occur by other means like birds or wind or even water. But we're going to see here that insects are probably the pollinators of the earliest flowering plants. They're still the most common, most diverse agents of pollination. And they're going to be specific to specific species of flowers that they're attracted to, and they are then adapted to be able to carry that pollen to other plants of that species and drop it off at the stigma that is awaiting. So even though we have this wide variety, there are some common features. So flowers have large, brightly colored petals that advertise the flower. Scent is secreted by the petals to advertise the flower. Pollen grains are large and spiky, so they stick to insects. They're attractive as a protein-rich food. Okay, the stigma is large and sticky to collect pollen from visiting insects. Okay, and then there are glands deep within the flower called nectaries that secrete a sugar solution that is attractive to insects as an energy source. Okay, and since they're so deep in there, insects can only reach them if they brush past the anthers and stigma, along the way picking up that pollen, and when they visit the next flower, being able to drop it off at the stigma of the next flower. Okay, now here we have another drawing skill. Okay, you should be able to draw a half view of a flower with all of these parts labeled. And note, just like the male and female reproductive system, you should technically be able to annotate your drawing with the names and functions of the flower parts. So here I have an example of what the drawing skill might look like without the functions listed. So I have the functions then here listed on the side. So go ahead, pause that, and make sure that you can identify all of those things. Okay, and then here I just have some strategies for creating biological diagrams. When doing drawing skills, there are a few things that IB would prefer. Just things like when you draw your line, don't put arrowheads on it when you're labeling something. Go ahead and pause this and read through them so that you correctly do your drawing skills. Next up, cross-pollination is transfer of pollen from anther in the flower on one plant to the stigma of a flower on another plant. This leads to fusion of male and female gametes from different plants, okay, which is great because we have genetic variation occurring then. We can have evolution occur, and this is essential at times of environmental change. Cross-pollination also promotes hybrid vigor. Okay, this just means that when you have genetically unrelated plants crossing, they tend to be very healthy. This may be due to having different alleles of many genes rather than just being homozygous and having two identical copies of a gene. Now, many plants, again, are hermaphrodite, like we mentioned earlier. Okay, they have both pollen containing male gametes and ovules containing female gametes, which then makes self-pollination possible. Okay, this is an extreme example of inbreeding where the male part of a plant would fertilize the female part of the same plant. Okay, however, there's a lot of problems with inbreeding. In humans, we have taboos or laws in most human societies that prevent members from marrying a close relative because it has long been known that such marriages tend to have a higher rate of miscarriages and disease. Okay, the reason that inbreeding is a problem is that it increases the chance of rare recessive alleles in one ancestor being inherited twice by an individual. Okay, inbreeding depression is a general term for premature death, failure to thrive, and infertility among offspring. Okay, and if you want to see a very clear example of inbreeding, okay, look at the European royal families, which ended with Charles II, King of Spain, who had his famous Habsburg jaw, okay, and a series of problems that happened due to all of this genetic inbreeding that we can see here in this family tree. So, mechanisms have evolved to reduce or prevent inbreeding in plant species, making self-pollination either less likely or impossible. So let's review a few of those strategies. Number 1, adaptations to facilitate transfer of pollen from one plant to another by an outside agent. As we've been discussing, insect pollinators, for example, allow for movement of pollen from one plant to another. Secondly, plants may separate anthers and the stigma styles or ovaries in separate male and female flowers on the same plant. Okay, or they could have those separations on different plants. Okay, did you know that papaya trees are either male or female? Okay, and finally, anthers and stigmas may mature at different times. Okay, which means that they wouldn't meet, that plant wouldn't be able to self-fertilize. So despite features of flowering that reduce the chance of self-pollination, it still occasionally can occur. And so in many plants then, this pollen just fails to germinate. The pollen tube stops growing before it reaches the ovary, preventing that inbreeding. Okay, this is called self-incompatibility. It's important because inbreeding decreases genetic diversity and vigor. Okay, self-incompatibility has evolved more than once. There are different mechanisms, but they all have a genetic basis. Okay, plants with the same self-incompatibility alleles cannot successfully pollinate each other. Okay, Dispersal and germination of seeds. So while seeds can't move themselves, they often travel long distances from the parent plant, called seed dispersal. It serves to reduce competition between offspring and the parent and spread the species. Depending on the structure of the fruit, the strategy might be that they are dry and explosive. They may be fleshy and attractive for animals to eat. They may be feathery or winged to catch the wind, or covered in hooks that catch onto the coats of animals. So lastly, for the SL content for this slideshow, let's contrast pollination and seed dispersal. So remember that pollination is the transfer of pollen from anther to stigma, most commonly by wind or animals. Whereas seed dispersal is transferring seeds, which are different than pollen. This is going to be our fertilized eggs from the female parent to a germination site, commonly by wind, animals, or explosion. That brings us to the end of SL content for D3.1. Let's go into more depth now with the HL content. Puberty is a sequence of developmental changes that form the transition between childhood and sexual maturity. Onset and progress through puberty is controlled by the brain via hormone secretion. And the key to this is gonadotropin releasing hormone, GnRH. The key word there is that gonadotropin. It's releasing the hormone that affects the gonads, which are the testes and ovaries. The hypothalamus synthesizes the hormone, secretes it into a blood vessel that carries it to the pituitary gland. Now, GnRH secretion starts in a fetus about 10 weeks after fertilization, and continues until the baby is 4 to 6 months old. But from then, it only resumes secretion when the brain determines the time for puberty has arrived, at some stage, usually during the teenage years, and then continues through puberty and adulthood. Gonadotropin releasing hormone's role is that it stimulates secretion of FSH and LH by the pituitary gland. In males, FSH stimulates testis growth, LH stimulates testosterone secretion by Leydig cells in the testes, and they both have roles in stimulating sperm production. And testosterone causes the development of secondary sexual characteristics in boys. In females, as we talked about, FSH stimulates the development of follicles in the ovary. The wall of the follicle secretes estradiol. LH stimulates development of the follicle wall into a corpus luteum after ovulation. The corpus luteum then secretes both estradiol and progesterone, and it's estradiol that causes development of secondary sexual characteristics in females. Now let's talk about gametogenesis, the generation of gametes, sperms and eggs, known as spermatogenesis and oogenesis. Male gametes are called spermatozoa, usually abbreviated to just sperm. Female gametes are oocytes, which are usually called eggs. Now in both spermatogenesis and oogenesis, we're going to have the same 4 stages: mitosis, cell growth, 2 divisions of meiosis, and then differentiation. However, there are significant differences in how these stages are carried out. So let's start with spermatogenesis. This happens in the testes, which are a coiled mass of seminiferous tubules with small gaps containing interstitial fluid between the cells. Those interstitial gaps are filled with those testosterone-secreting Leydig cells. The outer layer of cells in seminiferous tubules is the germinal epithelium, shown here. Now, what we're going to see is that we're going to see a process of mitosis and meiosis occur from the germinal epithelium towards the center of the seminiferous tubule. So let's begin with mitosis. And here we can see we have a diploid cell. It's going to produce 2 diploid cells through mitosis, identical to the parent cell. Now, of those 2 cells, one is going to go on then to do the 2 splits of meiosis, while the other one is going to go back and be able to regenerate this process again. So first, mitosis. Then, as the cells migrate inwards, they are going to go through a period of growth. They then divide by meiosis, which we know has 2 divisions. So we end up with 4 haploid cells. Okay, each of those haploid cells then go through differentiation into sperm by growing a tail, reducing their cytoplasm to a minimum. Okay, and this is going to be facilitated by large nurse cells called Sertoli cells that are seen here. Okay, so here is an outline of what we just talked about. Feel free to pause and read through that. Here's another image, and we can see again the mitosis occurring. Okay, and then towards the center, we eventually end up with sperm. Now let's contrast that with oogenesis. This happens in the ovaries. The first stages are completed before birth. The outer layer of cells in the ovaries of the female fetus is the germinal epithelium. So again, we begin with mitosis. Okay, similarly, mitosis has one parent cell becoming 2 identical daughter cells. Those cells migrate inwards to distribute themselves throughout the cortex of the ovary. Okay, but then when a fetus is 4 or 5 months old, those cells start the growth phase. At the same time, they start to divide by meiosis. We're going to see a pause. By the 7th month, they are still in the first division of meiosis. They're surrounded by a single layer of cells. Okay, and that is now called a primary follicle. That egg that is going to be surrounded by a single layer of follicle cells. There are about 400,000 primary follicles in the ovaries at birth. They don't undergo further development until after puberty. No more primary follicles are ever produced. Then, at the start of each menstrual cycle, a small batch of primary follicles is stimulated to develop by that FSH, follicle stimulating hormone, and usually only one goes on to become a mature follicle containing an egg. Now, note the difference. While we have mitosis and growth at the beginning of oogenesis, those 2 divisions of meiosis, instead of producing 4 haploid gametes, only produce 1. Because after each division of meiosis, you'll see here this primary oocyte is going to split and have 1 secondary oocyte produced, but then also a polar body, which just breaks down. What this does is it allows for more of those organelles and food storage to be really concentrated in the secondary oocyte, and that process repeats again for us to eventually form this ovum. A second polar body is produced in that second round, which again, all the good stuff kind of goes to the ovum, and the polar body can break down. Okay, so here's the cycle again if you'd like to pause and read through it in a different format. All right. Let's contrast then spermatogenesis and oogenesis. In spermatogenesis, we're going to have 4 gametes from 1 meiosis. In oogenesis, just the 1 because there were also 2 polar bodies produced that get broken down. Okay, whereas spermatogenesis can generate millions of sperm per day, oogenesis only about 1 per month. Okay, sperm are released during ejaculation. Oogenesis are released at ovulation on about day 14 of the menstrual cycle. Okay, sperm are produced from puberty onwards, taking about 75 days. Oogenesis, the process is initiated while the female is still a fetus, okay, but is then completed during the menstrual cycle, released on about day 14, occurring from puberty onwards till menopause. Now remember, sperm have little cytoplasm helping them to swim faster. Eggs contain more cytoplasm than any other human cell to provide food reserves for the embryo, and in fact, eggs are the largest human cell. Okay, now remember that if 2 sperm were to penetrate an egg at the same moment, a triploid zygote would be produced and it would soon die. It is inviable. So it is very important that only a single sperm can penetrate the egg. So let's talk about the 2 reactions that occur in order for this to make sure that this is what happens. Now, first up, the sperm has to get in. That's going to happen from the acrosome reaction. Okay, now remember that there is a acrosome containing digestive enzymes in the head of a sperm. Okay, there is a zona pellucida, which was that coat of glycoproteins seen here. Okay, it surrounds and protects the egg. The sperm are going to bind to specific glycoproteins in the zona pellucida. Okay, that's going to trigger release of those enzymes in their acrosome, which then begin to digest the glycoproteins, weakening the zona pellucida, which then allows the sperm to enter into the plasma membrane and fuse with it and release its nucleus. But remember, we only want one. So then we have the cortical reaction. This is going to prevent other sperm from being able to pass through, making the egg then inviable. Now, near the edge of the egg are called cortical granules that are filled with enzymes. Okay, they're vesicles that when the first sperm penetrates, that activates the egg. Okay, the egg moves the cortical granules and does exocytosis, releasing the contents. Okay, and those enzymes that are released cause a general toughening of the zona pellucida, okay, making it impenetrable to any future sperm. Okay, the enzymes also change specific glycoproteins, okay, and so sperm can just no longer bind, preventing polyspermy, many sperm from fertilizing a single egg. Okay, so here we now have a zygote. We had 2 haploid nuclei, okay, a haploid nuclei from the sperm and one from the egg. We've now fused them through fertilization and produced a zygote. What happens next? Well, we're going to have replication of DNA, okay, and that's going to start about 6 hours after fertilization and takes about 3 hours to complete. Okay, the 2 nuclei undergo major changes in the pattern of gene expression. Okay, about 30 hours after fertilization then, they end up dividing jointly by mitosis, okay, providing 2 genetically identical diploid nuclei, which separate when the cell divides. Okay, then during the following days, that embryo undergoes rapid rounds of the cell cycle. The number of cells doubles about every 18 hours. Okay, but we aren't getting any new nutrients during this entire period, which takes about a week, as that fertilized egg continues to move down the oviduct towards the uterus. This is why it's so important that the egg was so large, because all of these divisions that are occurring now, those cells are getting smaller and smaller, okay, because they're still just within the same, boundaries and the same amount of food and nutrient sources that they started with. Okay, so while initially that embryo was a solid ball of cells, at 6 or 7 days old, it is changed to a hollow ball called a blastocyst. Okay, it's got an inner cell mass, which is going to become the new human. Here's the toughened zona pellucida, thanks to that cortical reaction. Okay, there's just some fluid inside the blastocyst. Okay, now it's moving down the oviduct. Okay, over a period of about a week, approximately on day 7, the embryo will start implantation, okay, in the endometrium, which is that uterine lining. Okay, and at this time, the blastocyst has used up all the reserves of the egg cell. It needs an external supply of food. So it's going to attach, okay, to the endometrium in a process called implantation. The outer cell layer of the blastocyst has finger-like projections that grow into the endometrium and are able to exchange materials with the mother's blood, including absorbing foods and oxygen. Okay, and that inner cell mass grows and develops into a human body. By 8 weeks, it started to form bone tissue. It's now called a fetus rather than an embryo. The new individual is recognizably human and will soon be visibly either male or female. Okay, back to HCG, which we mentioned at the beginning of this slideshow. It's a protein produced by the embryo's trophoblast cells from the blastocyst stage onward. Continuity of HCG production is essential to maintain pregnancy and avoid miscarriage. Okay, so what does it do? In the first 10 or so weeks, HCG stimulates the corpus luteum to develop and then to secrete progesterone. This hormone helps to support the pregnancy by preventing degeneration of the uterine lining. Remember, that's progesterone's primary job, is maintaining the endometrium. Then, after about 8 to 12 weeks of pregnancy, the placenta starts to secrete progesterone in response to HCG. The corpus luteum stops hormone production and breaks down. Trophoblast cells in the placenta continue to secrete HCG throughout the remainder of the pregnancy, continuing to stimulate progesterone secretion by placenta cells and therefore maintenance of the endometrium throughout the pregnancy. Okay, so once again, I mentioned earlier, pregnancy tests are based on detection of HCG in a woman's urine using monoclonal antibodies that bind to HCG. If you'd like to know more, okay, you can pause the video and read this summary. Now, humans are placental mammals. Okay, there are two other groups. There are monotremes, which lay eggs, like a platypus, and there are marsupials, which give birth to relatively undeveloped offspring that develop inside a pouch, like a kangaroo, as seen here. Now, by the stage that a marsupial would be born, a human fetus has developed a relatively complex placenta, so it's able to remain in the uterus for months longer. The placenta is needed because the body's surface area to volume ratio becomes smaller as the fetus grows larger. So let's look at the placenta a little bit more closely. Now, the placenta is actually made of fetal tissues, okay, in intimate contact with maternal tissues in the uterus walls. The fetus develops membranes that form an amniotic sac containing amniotic fluid. The basic functional unit of the placenta, okay, is a finger-like piece of fetal tissue called a placental villus, shown here in yellow. These villi together provide a large surface area. They increase in number during pregnancy to cope with the increasing demands of the fetus for exchange of materials with the mother. Now, the maternal blood is going to flow in these spaces around the villi. Okay, fetal blood then circulates in blood capillaries that are close to the surface of each villus. The distance between fetal and maternal blood is very small. Okay, however, they don't mix. Remember, a fetus and a mother might have different blood types, right? If the blood was to mix, it would be recognized by the other as non-self, and an immune response could be mounted. And so while the blood is very close to each other, it doesn't mix. Okay, instead, we're going to see that nutrients and waste products and dissolved gases are going to move back and forth across this placental barrier. Okay, so here's a summary of what goes in which direction. Okay, maternal blood is going to provide oxygen, glucose, antibodies, and water to the fetus. The fetal blood then is going to return back carbon dioxide, urea, and potentially water, depending on how the balance is going through osmosis. You should know, okay, about what type of transport is used in each of these and why. For example, oxygen is going to move through diffusion because it's very small. Okay, glucose through facilitated diffusion because it's polar. Antibodies are going to move through endocytosis because they're so large. Okay, by about the 9th week of pregnancy then, the placenta has started to secrete estradiol and progesterone in large enough quantities to sustain the pregnancy. The corpus luteum is no longer needed. Okay, so we're going to have a switchover, and this is a dangerous time for miscarriage if the switchover fails. During pregnancy, progesterone inhibits secretion of oxytocin by the pituitary gland, okay, and inhibits contractions of the muscular outer wall of the uterus called the myometrium. So at the end of pregnancy, hormones produced by the fetus signal the placenta to stop secreting progesterone, so then oxytocin starts to be secreted. Okay, now oxytocin is what stimulates contractions of the muscle fibers in that myometrium. Okay, these contractions are then detected by stretch receptors. Those then signal back to the pituitary to increase oxytocin secretion. By increasing oxytocin, those contractions become more frequent, more vigorous. This is an example, our classic example of positive feedback. Okay, oxytocin stimulates stretch receptors. Stretch receptors then stimulate the pituitary to create more oxytocin. And so we just see stronger and stronger contractions, which then eventually lead to the baby being able to be born. Okay, and that looks like relaxation of muscle fibers in the cervix, causing it to dilate. Uterine contraction bursts the amniotic sac. Amniotic fluid passes out. Further contractions finally push the baby out through the cervix and vagina. The umbilical cord is broken. The baby takes its first breath and achieves physiological independence from its mother. Okay, very last thing for this slideshow, hormone replacement therapy and the risk of coronary heart disease. This is actually really interesting. Now, first up, what is it? Hormone replacement therapy is a treatment used to relieve menopausal symptoms. It supplements the levels of the hormones estrogen and progesterone that would naturally decrease as a woman approaches menopause. Okay, it can relieve symptoms such as hot flashes, night sweats, etc. It can also help prevent osteoporosis. Okay, but here's what's interesting. Okay, it's been suggested that hormone replacement therapy might increase the risk of coronary heart disease. Now we're going to look at how correlations may be based on a causal relationship, but correlation doesn't prove causation, and this is a very important principle in science. So let me explain why. In early epidemiological studies, right, epidemiology is the study of the spread of diseases in human populations, it was argued that women undergoing hormone replacement therapy had a reduced incidence of coronary heart disease. This was deemed to be a cause and effect relationship. Later, randomized controlled trials showed that hormone replacement therapy actually led to a small increase in the risk of coronary heart disease. Turns out the correlation between hormone replacement therapy and decreased incidence of coronary heart disease is not actually a cause and effect relationship. The initial fallacy of a causal relationship was coincidental because patients who can afford hormone replacement therapy tended to come from groups with a higher socioeconomic status. And a higher socioeconomic status is what is thought to be causative of that decreased rate of coronary heart disease. So it's not actually replacing hormones that led to that decreased rate of coronary heart disease. It's just the socioeconomic piece that led to the fallacy of it appearing to be a cause and effect relationship. So correlation doesn't prove causation. Let's move on now to D3.2 Inheritance. First up, gametes. Those sex cells, eggs, and sperm are haploid. They contain one chromosome of each type. Okay, whereas the somatic or body cells or diploid, they have two chromosomes of each type. In humans, there are 23 pairs in a somatic cell, just 23 chromosomes in a haploid gamete. So if we want to investigate inheritance and actually do genetic crosses, that can be done using, for example, a paintbrush, where you can actually use a brush to gently pick up the pollen from an anther, okay, and then dust it onto a stigma of the plant that you would like to cross with the other plant. Okay, it is important though that we don't allow other sources of pollen to land on the stigma, so we know that it is the actual pollen from our paintbrush that was used to fertilize that flower. And so that can be done by cutting off the anthers of that flower so it doesn't self-pollinate, and then enclosing that flower in a paper bag to prevent any additional pollen from being able to land on that stigma. So when we do a cross, okay, we're going to start with the parental generation, our P generation. We then cross, and that first generation is called the F1 or the filial 1 generation. Okay, and then the generation after that, when we cross those offspring, is going to be the F2 generation. Now, the father of inheritance and genetics is Gregor Mendel. Okay, he studied pea plants and he looked at 7 different traits, and he grew thousands of these plants and calculated ratios and made a lot of discoveries of some basic principles of inheritance. So we can use a Punnett grid to analyze the results of genetic crosses. Okay, and we'll talk about what each of these different uppercase versus lowercase letters mean, what it means for the genotype versus the phenotype. Okay, but first we need to understand that there are different alleles of a gene. Remember, a gene is just a length of DNA that codes for a specific trait. And so we can have variations of those different genes, and we call those alleles. They can be as similar to each other as just a single base difference, okay? Or there could be large sections of that length of DNA that is different. Now, humans and other diploid organisms have 2 alleles of most genes, one inherited from each parent. Okay, so let's look at an example. If we, for example, had 2 big B alleles, so these would be the dominant form indicated by the capital letter, Okay, this would code for normal wings. This would be one gene from mom, one gene from dad, but the same allele at this time location for the wings. If you have a big B and a little b, a dominant allele from one parent and then a recessive from another, that would be considered heterozygous. Hetero means different, homo means the same. Heterozygous, if you have a dominant and a recessive allele, only the dominant allele is going to show up in the phenotype or the physical representation of what that gene then codes for. And then here we can see our last example where we have homozygous recessive alleles, those lowercase. And so that's in this case going to code for wrinkled wings if you get that recessive allele from both parents. So these here represent the genotypes. Okay, then here is the phenotype, the physical, just think phenotype starts with PH, so does physical, representation of what the genotype ends up coding for. Okay, so if a parent is homozygous, then we'll see through the process of meiosis, which again is going to start with DNA replication and then go through 2 divisions. Okay, every single haploid gamete produced is always going to have, in this case, that big B. Same if the parent was homozygous for little b, all of the time their haploid gametes produced will be little b. However, if the individual is heterozygous, then note half of their haploid gametes produced will be big B and half will be little b, and there will be that exactly 50% chance, okay, of which of those gametes is produced. Okay, so again, the genotype is just going to be those alleles and they code for the phenotype. Here you can see that heterozygous condition, the dominant allele will be completely dominant over the recessive allele, and the phenotype in this case then will be purple, okay, which is indistinguishable from an individual with the genotype big B, big B. Now, traits can either be just genotype that actually controls them. For example, the blood type, the ABO blood groups are going to be just based on genotype. Sometimes it's based on environmental interacting with genotype. Things like height in humans is coded for in the genotype, but then also environmental factors like nutrition are going to play into the height that the individual actually gets to. And then there are some traits that are environment only, things like scars, language. Okay, those traits are all based simply on environment. Okay, so here is a picture of one of the pea plants that Gregor Mendel studied. He can either cross individuals using the methods described earlier, or they can be allowed to self-pollinate. Okay, and he started by developing pure breeding varieties that always produced offspring with the same traits, and therefore those were the homozygous individuals. Now, one might think that if Mendel was to cross a tall pea plant with a dwarf pea plant, that you might end up with a pea plant with an intermediate height. However, height in pea plants is controlled by just 2 different alleles, and so the actual results that Mendel discovered was that tall plants crossed with dwarf plants, that F1 generation had all tall plants, no intermediates. And so, as in further investigation, okay, Mendel discovered that while all that first generation were tall pea plants, if he then crossed individuals from that first generation, he got a 3-to-1 ratio of tall to short. That short pea plant phenotype showed up in the F2 generation. And so we can represent what happened using this system of letters. Okay, we're going to use capital letters to indicate the dominant allele. So the pure breeding tall pea plant would be big T, big T. We're going to use the lowercase t to represent the recessive alleles. So the pure breeding dwarf pea plant is little t, little t. And individuals then of that first generation are going to be heterozygous, big T, little t, because this parent is always going to give a big T, this parent always gives a little t. So all individuals of that next generation are big T, little t. Okay, Then if we cross individuals of that generation, we're going to see this 1 to 2 to 1 ratio of genotypes. When crossing, we're going to have 1 big T, 2 heterozygous big T little t's, and 1 homozygous recessive little t. Okay, now while we have a 1 to 2 to 1 ratio of genotypes, we have a 3 to 1 ratio of phenotypes. 3 of those individuals will be tall and 1 will be dwarf. All right, this is an example of a monohybrid cross where we're just looking at one character, in this case, height of the pea plant. Okay, so follow this image. Okay, make sure you understand exactly what's happening with the parental F1 and F2 generations. All right, now organisms can respond to their environment by varying their patterns of gene expression and therefore their traits. Okay, this is known as phenotypic plasticity. It's particularly useful if the environment a population inhabits is heterozygous rather than uniform. Okay, it's not due to genotype, it may be reversible. For example, a person with pale skin may become darker skinned if there's an increase in the intensity of sunlight. Okay, a change in gene expression results in increased synthesis of the black pigment melanin in the skin. Okay, known as a tan. If the sunlight stimulus diminishes, gene expression reverts to its former pattern. The skin gradually becomes paler again as melanin concentration reduces. Phenotypic plasticity. Okay, an example that you should be familiar with is phenylketonuria. This is a human disease and it's due to a recessive allele. And so, the only way to develop this disease is if you receive the recessive allele from both parents. Now, if both parents are carriers, neither one will have the disease, okay, and they will likely be unaware that they are carriers. And so, of their offspring, there will be a 1 in 4 chance that their offspring would have this disease. Okay, now what is the disease? It is a mutation of the gene that codes for an enzyme that converts phenylalanine into tyrosine. Now, both of these are amino acids that are used to make proteins. And so, if you can't make tyrosine because you don't have the gene to make this enzyme, okay, you're going to end up with a accumulation of phenylalanine in the body and a deficiency of tyrosine. This can impair brain development, leading to intellectual disability, mental disorders. Okay, this can be prevented by screening for PKU at birth. Okay, and if it's a positive result, then the child would be given a diet low in phenylalanine, okay, which would be the treatment for this condition. A gene pool is all the genes of all the individuals in a sexually reproducing population. You can see here that in this particular gene pool, we have 3 different alleles. Okay, these alleles again are just different forms of a gene. They can be as similar to each other as one base difference. Anywhere that there is a single nucleotide polymorphism or a difference between alleles, okay, we call a SNP. Okay, and there can be just a single SNP between different alleles or multiple. Okay, another example that you should be familiar with are ABO blood groups. This is a special case where we have multiple alleles, as seen in that earlier example of the gene pool. There are 3 different alleles, okay, that are possible. Each individual will only receive 2, one from mom and one from dad, but there are 3 options. Okay, those options include, okay, the 2 codominant alleles, big IA and big IB, and then there are sets of allele, little I. And you can see here that then there are 6 different combinations that are possible from those 3 alleles. If an individual gets 2 big IAs, okay, they're going to be type A blood. Same if they get a big IA and a little I, because that's recessive, we're just going to have, again, type A blood. The same pattern happens for group B, big IB, group B, and then if there's a recessive allele, still group B. Okay, if the individual receives 2 recessive alleles, little I, we call that blood type O. And because this is a codominant system, we have 2 dominant alleles. Okay, this is a special case where if an individual gets big IA and big IB, they have this special blood type called blood type AB because neither of these alleles are dominant over the other, and so we call that codominance. Okay, and you can see here, you should be familiar with this system, that somebody that has blood type A is going to produce antigen A on their red blood cells. Okay, in their plasma, the liquid part of their blood, they'll have anti-B antibodies. And so they cannot have B or AB blood because those individuals would have antigen B. Okay, and their antibodies in their blood would attack those red blood cells. They can receive only blood from someone else with blood type A or someone with blood type O. Okay, that is a universal donor because they don't have any antigens on their red blood cells that would respond to antibodies in the blood of any of the other individuals. Okay, and you can then go through and follow that pattern throughout. You should be familiar with this system. Okay, so codominance, again, then a great example of codominance in flowers is that if an allele is codominant, in this case, both of those forms, okay, are going to show up. For example, if this is red flower breeding with a white flower, all the individuals are going to be heterozygous and they're going to show both red and white parts of the flower. In contrast, incomplete dominance is when you would have a red and a white flower. And instead of red and white flowers, we see an intermediate phenotype, which is pink. So this is incomplete dominance, and you should be able to differentiate that from codominance, where both traits show up. Okay, sex is determined in humans by the 23rd pair of chromosomes, okay, which are either X or Y chromosomes. Now, the X chromosome is much larger than the Y chromosome. Females are going to have 2 X chromosomes. Males will have 1 X and 1 Y. Okay, and this is why we have a 50-50 chance of producing male and female offspring. A female is always going to either give an X or an X with a 50% chance. Okay, a male is going to 50% of the chance give an X sperm and 50% of the time give a Y sperm. And so you can see when we cross those, 50% of the time the offspring will get an X from each parent, and 50% of the time they'll get an X from mom and a Y from dad, and therefore become male. Therefore, turns out the father 100% of the time is who determines the sex of the offspring. It is always determined by the sperm. Okay, now while this XX and XY are the standard inheritance, know that there are also some sex chromosome abnormalities. You don't need to be familiar with these in detail, but just know that they exist. For example, an individual with 2 X chromosomes and a Y develops as a boy with Klinefelter's syndrome due to non-disjunction, and you can see here what the karyogram would look like for that individual. Okay, another example would be an individual with 1 X and no other sex chromosome due to non-disjunction would develop as a girl with Turner syndrome. Okay, so how does sex determination occur? Okay, at just about 5 weeks old, a human embryo will start to develop a pair of gonads. Now, initially, they could become either testes or ovaries. Now, on the Y chromosome, there's a gene called the SRY gene. Okay, it's known as the testis determining factor. If it is present, okay, it's going to start, those developing testes start to secrete testosterone, okay, and a male develops, okay, and in the absence of that TDF gene, the fallback position is developing into ovaries and that individual becomes female. Okay, so we are then going to see a different inheritance pattern for genes that are carried on the X chromosome, and we call this sex linkage. Okay, it was investigated in fruit flies. Fruit flies are a great laboratory organism for investigating genetics because their life cycle is only about 2 weeks. And so we're able to very quickly do crosses and look at the results of those crosses and clearly be able to see how different traits are inherited. And so while most crosses in fruit flies don't show sex linkage because they're on the autosomes, the non-sex chromosomes, we are going to see that in the particular red eye versus white eye trait, that is carried on the sex chromosomes. And so we're going to see then this inheritance pattern. Okay, now note if we have a female who is a red-eyed, which is the dominant trait indicated here by big R, crossed with the male who is white eyes, indicated here by this recessive trait little r, we're going to see that 100% of the offspring have red eyes because that X big R will be given to every individual and it's dominant. 100% of them have red eyes. Now, on the other hand, if the female was the white-eyed individual, okay, and the male was the red-eyed, we're going to see here that the female is always going to give this recessive X little r. Now, the male is going to give the X big R. That X only, though, gets passed on to the daughters because the sons are going to get the Y. And so the sons are only going to get an X chromosome from mom, and since mom had the recessive little r, they're going to have white eyes, whereas all of the daughters will have red eyes. Okay, now note a couple things. This letter here indicated in superscript, whether it's on a sex-linked chromosome or if it's just a normal Mendelian cross, usually that letter is chosen by being the first letter of the dominant trait. So since R here is the dominant trait, red eyes is the dominant trait, we're going to choose R. Note that when doing sex-linked inheritance then, that's going to be indicated in superscript, okay, on the X for representing the X chromosome. Okay, so here is just an example of what that's going to look like then for all of those different particular forms. Now, we don't want to confuse sex linkage with gene linkage. Gene linkage is just when two or more genes are located close together on a chromosome, so they tend to be inherited together. Okay, it can be on any chromosome. And so it's actually possible for a gene to be both a linked gene on the sex chromosome and sex-linked if they're located close together on the X chromosome. Okay, now you should be able to look at a karyogram to identify whether an individual is male or female. You're going to look for the very last set of chromosomes. In this case here, our last set of chromosomes has one long and one short. We know that X is long, Y is short, and so this individual should be identified as a male. Here we have two long chromosomes. They're actually labeled in this case, so this individual should be recognized as a female that has two X chromosomes. Okay, another example of a sex-linked genetic disorder is hemophilia. This is a recessive condition, okay, and it results in the lack of a clotting factor, and so blood doesn't clot normally. And so if this individual has a cut, it's going to take much longer than normal for the bleeding to stop. Okay, and internal bleeding is also very common. So looking at the same exact patterns that we just reviewed in fruit flies, you should be able to predict the inheritance of hemophilia. Okay, now interestingly, because there's only a 1 in 100 million chance of girls getting hemophilia, the actual results of how many girls get it is even lower than that. Because hemophilia is clear from a young age, a male with hemophilia would have to make the decision to risk passing on the condition to his children, and so that risk is even lower. Okay, whereas it does show up in males because a non-hemophiliac father may basically have offspring with a carrier female that wouldn't know that she would then be passing that on to half of her male offspring. Okay, another example is color blindness. Okay, if you can't see any of the numbers that are indicated in each of these 6 circles, okay, you could potentially be colorblind, and it also follows this same pattern. So I would pause this, read through this question, okay, make sure that you can indicate what's going on here in this practice problem. Now, note any sex-linked individuals, if you're asked for the phenotypes or the genotypes of the offspring, make sure that you differentiate between the female and male offspring. Okay, here's another practice question. Feel free to pause. And a final practice question. All right, so medical research has already identified more than 4,000 of these genetic disorders, okay, and we can track them using pedigree charts, these family tree diagrams, and there are some standard conventions, including that males are shown as squares, females are shown as circles. Okay, anything that is shaded or crosshatched indicate an individual affected by the disease. Parents and siblings are linked with horizontal lines, parents and their offspring with vertical lines. Okay, Roman numerals indicate different generations, and then these Arabic numbers are used for the individuals in each generation. Okay, let's look at an example of how hemophilia has passed through the British royal family. We're gonna, you might recognize over here, for example, Queen Elizabeth, who just passed away a few years ago. Prince Charles is now King Charles as of the making of this video. But we're gonna look over here at some of their relations in this part of the family tree, also descended then from Queen Victoria. Okay, and this is a great little practice question here. Okay, I'm going to go ahead and just put the answers up, but you should be able to go through and determine the potential genotype or the for sure genotype of any of these individuals. So this is what you should be able to come up with. Okay, so how do we deduce patterns? Some tips. First of all, determine if the trait is dominant or recessive. If it's dominant, one of the parents must have the trait. If you see that there are two unaffected parents with an affected offspring, then it has to be recessive. Okay, determine if the chart shows an autosomal or a sex-linked trait. Okay, for example, in sex-linked traits, males are going to be much more commonly affected than females. Okay, in this case, we have pretty equal numbers of males and females, and so we know it's going to be an autosomal rather than a sex-linked trait. Okay, here's a couple of examples. You can pause and read through. Example 1 and example 2. All right, variation is one of the defining features of life. Two individual organisms in the same species have fundamental features in common that are shared by that species, but are also likely to differ in many traits. Okay, and there are two types of variation, either continuous or discrete. Okay, continuous variation is where we have a range of types along a continuum. Okay, the spectrum of visible light is a continuum. In this case, the trait is influenced by multiple genes. Okay, environmental factors may influence the trait. Examples include skin color, tree height, body mass of animals, human wrist circumference, right? We don't either all have dark or light skin. Our skin is along a continuum of skin colors. On the other hand, discrete variation is when there are no intermediates. There are a few possible phenotypes. Okay, there is usually just going to be one or at most a few genes if there's a genetic cause. Usually environmental factors do not influence the trait. Examples include those ABO blood groups that we talked about earlier, number of eggs laid by birds, or left or right-handed snail shells. Okay, so skin color in humans is, again, partially due to the environment, okay, and partially due to genetics. Okay, this discrete data is countable. This is an example here of some discrete countable data versus this is an example of continuous data, corn cob length, and you can see here this normal distribution. Okay, you should be familiar with calculating mean, median, and mode. If you are not, you should pause this screen and read through this at this time. Okay, this is an example of a box and whisker plot. You should know that the center of a box and whisker plot is the median. Okay, we then have the upper and lower quartile indicated by the box. Okay, and then the maximum and minimum indicated at each end of what's called the whisker. Okay, here's some information about how to calculate the interquartile range. If you don't already know how to do this, you should pause the video at this time. Okay, here's a practice problem from the textbook. Okay, go ahead and take a look at that. Pause if needed. Here are the answers. Second practice problem with the answers. Third practice problem. Answers. And lastly, there we go. All right, this brings us to the end of SL content for D3.2. Okay, we're going to move on now to our last part of the HL content for this video. Segregation is separation of alleles in the gene so that one allele is inherited but not the other. Independent assortment is the segregation of the alleles on those two genes, so the outcome with each gene has no effect on the other. It is 100% 50-50 chance of which of those alleles gets passed on to the offspring. Okay, segregation and independent assortment are the consequences of events in meiosis. Okay, and it just has to do in this case with which pole each of the individual sets of homologous pairs, which side they line up on when they line up during metaphase. Okay, and that again is a 50-50 random chance. Okay, there's no influence on the orientation of other pairs of chromosomes. Each one has that 50-50 chance of how they line up. Okay, now we know that some genes don't assort independently like this because they are located then on the same chromosome close to each other, and so they tend to be inherited together. The only time they wouldn't be is if crossing over occurs and we have a flip-flop where we exchange genetic material, and so in this case, those genes may not be inherited together if crossing over occurred. Now, monohybrid crosses test the inheritance of a single gene, but we can also do dihybrid crosses to investigate the joint inheritance of two genes to see if they get inherited together. Okay, so let's take a look at an example of a dihybrid cross of pea plants where we look at both the smoothness versus the roughness of a seed and the yellow versus green color of a seed. Now, if we're given that smooth is dominant over rough and yellow is dominant over green, then if performing a dihybrid cross, okay, we would be able to see, okay, the following potential genotypes that would lead to each of these four options for the phenotypes. So when doing a dihybrid cross, the first thing you want to do is you want to FOIL. If you remember this from math class, okay, that's where you're going to do a cross and you want to look at every possible cross. And so you cross first. FOIL stands for first, outside, inside, last. So first you cross the first, the big S and the big Y. Then you do the outer, big S, little y. Then you do the inner, little s, big Y, and then you do the last. So FOIL. Okay, you come up with the possible gametes that can be produced from this particular genotype. And you do the same thing for both parents. Okay, next you are going to lay out each of these four genotypes on one side of a Punnett square that is going to be a large Punnett square that has 16 potential offspring, and you'll do the same thing with the gametes of the other parent along the top. And then you will just do a cross, okay, and you will determine your 16 potential offspring, okay, and then you can differentiate between them. Notice here that we can say we're going to find this classic 9:3 to 3:1 ratio. Okay, and this is what happens if you cross two parents that are both heterozygous for both of the traits in a dihybrid cross. You will always get this 9 for both dominant traits, 3 and 3 for having one dominant and one recessive of each of the traits, and then 1 out of 16 with both of the recessive traits. Okay, then here's another example then. If you're asked to predict the ratio of phenotypes if you are crossing a heterozygous individual with a pea plant that is homozygous recessive for both traits, okay, you would actually get this 1:1 to 1:1 ratio. Okay, go ahead and pause if you want to look and follow how that works. Okay, note that these ratios for dihybrid crosses only applies if genes are on different chromosomes or are far enough apart on one chromosome for recombination rates to reach 50%. Okay, there are about 20,000 genes in the human genome. Each of those genes has a characteristic sequence, okay, and it also has a specific location called the locus. Okay, it's either going to be on one of the 22 types of autosome, numbered 1 through 22 in decreasing size. 1 is going to be the longest chromosome, 22 is the shortest, or on the 23rd pair, which is the sex chromosomes, X or Y. Okay, you should be exploring genes and their polypeptide products in databases. Okay, you should find genes with loci in different chromosomes and in close proximity on the same chromosome. Okay, so we've already covered this. If genes are close together, they tend to be inherited together. We call those linkages. Okay, and if they are linked, we're going to see a specific pattern that is different than if they are not linked. Okay, for example, if you see this type of ratio where you have a large amount of two particular individual phenotypes, and small amounts of these, then you can determine that these are linked genes. And we have some crossing over that has occurred in these smaller amounts of individuals. Okay, those are known as recombinants when crossing over has occurred. And you can see here that they have a specific linkage of way of indicating them. We write them like this, where each of these vertical lines represents the chromosome. And here you can see the allele indicated on the chromosome and the letter indicating whether it is positive or recessive. Okay, so let's look at an example. These here are going to be individuals that are not experiencing recombinance when these two individuals are crossed. However, when recombination does occur, right, and there's a, say, for example, crossing over and say little l and big L are switched here, then we can potentially get smaller numbers of these individuals. Okay, here you can see an example of the Drosophila, the fruit fly genome, and you can see that there are these different loci on their different chromosomes that are going to code for the different traits that the fruit flies have. Okay, oh, and here on the X chromosome with those white eyes, like that problem we looked at earlier where eyes were red or white. Okay, so what we can actually do is to map the genome, we can look at how frequently those recombinants occur in dihybrid crosses, and therefore that shows how far apart pairs of genes are. We can create maps of those genes. Okay, and now that we have the ability to actually sequence the genome, okay, we can bypass that sort of cumbersome method of having to do trials and trials and trials of fruit fly crosses and counting the number of recombinants that are produced. Okay, so again, we're going to use this notation when indicating whether we're going to be having recombination occur. Each of these, just think of this as the chromosome, one from mom, one from dad. These are the alleles from mom, these are the alleles from dad. And so we know then if crossing over occurs, okay, we have a flip-flopping of these two alleles. Now, in this case, of course, if it's homozygous, it makes no difference if they cross over. It's only going to make a difference if we have a heterozygous individual with one allele being different from the other. Okay, so then any organism that has experienced crossing over, we call that a recombinant. Okay, here's an example. You can read through if you'd like to pause the screen. A second example. All right, let's talk about the chi-squared test. This is a commonly used statistical hypothesis test. Okay, it's going to test goodness of fit. So how well does a prediction from a biological or statistical model fit the observed values? Note that we also did a chi-squared test when we did ecology, okay, to study whether two particular species tended to be located at the same location together or independently. So we can also use this to determine if an expected ratio based on a Mendelian cross is what is actually observed. So we're going to start by setting our possible hypotheses. Our null hypothesis says that the traits fit the predicted ratio. Our alternative hypothesis says the traits don't fit the predicted ratio. This is going to be our alternative hypothesis and indicate that crossing over is likely occurred. So the first thing you want to do is state those hypotheses. Then we're going to draw up a contingency table of the observed frequencies. We're going to calculate our expected proportion of individuals with each phenotype. We'll determine the degrees of freedom by just looking at the number of classes and subtracting 1. Okay, we'll find the critical region by looking at a table of chi-squared values, and then we'll use our chi-square equation to calculate our chi-squared value and compare it to our critical value. Okay, once we compare it, we'll be able to determine that if the chi-squared value is not in the critical region because it is equal to or below the value obtained from the table of chi-squared values, okay, we fail to reject the null hypothesis. Okay, there's no difference between predicted and observed values. Okay, if, however, the calculated value is in the critical region, we reject the null hypothesis. Okay, there's significant evidence that the Mendelian ratios do not describe the proportions of each phenotype in this case. So let's look at an example. Here we're going to have two different chickens breeding. Okay, we're going to find out that all of them in the expected ratio is 9 to 3 to 3 to 1. We've got two different heterozygotes that are being bred. Let's use a chi-square analysis to determine, okay, if some numbers that were given fit the expected ratio. So the first thing we're going to do is we're going to just state our null versus our alternate hypothesis. Okay, and then we're going to use our 9 to 3 to 3 to 1 expected ratio to calculate with our total number of individuals that were given in the problem of 190, how many do we expect to be in each of these four characteristics based on the 9 to 3 to 3 to 1. Next, we're going to determine our levels of degrees of freedom. There are four different classes, so we just do 4 minus 1 and we get 3. You will then always be provided the critical value table. You do need to know that you would look for the 0.05 level of significance. And then you would look at your degrees of freedom. In this case, we calculated it to be 3. So our critical value would be 7.815 as found here in the table. Next, we're going to do our chi-square analysis. We're going to take each of these expected numbers that we calculated a second ago, and we're going to compare them to the observed values that are given in the equation. And we're just going to use this chi-square equation, which you should memorize, which is the sum of the observed minus expected squared value divided by expected for each of these four. And we're going to add all of those up. So we're going to take the observed, okay, so in this case, 111 white pea. Okay, we're going to subtract the expected, which was 106.9. We're then going to square that value, then divide it by that expected value again. And we're going to do that for each of these four sets and add them all up. Okay, and it'll look like this. Okay, then our chi-squared value indicated by this capital X shape, which is the Greek letter chi, squared is 1.56. Now, the chance of I be asking you to actually calculate this out is fairly small. You should still be able to do it, but more likely they're going to ask you a question where they give you what the chi-squared value is. Okay, they might have you find the critical value, but either way, they might ask you to then draw the conclusion of what can you conclude based on these two numbers. Now, what you should look for is whether it's greater or less than the critical value. And in this case, our chi-squared value is less than the critical value. Therefore, it's outside the critical region. Okay, we have to reject the alternative hypothesis, accept the null hypothesis that there's no significant evidence that 9 to 3 to 3 to 1 is unsuitable. This suggests that independent assortment is occurring. The two genes are unlinked. And so the opposite would be true. If the chi-squared value is greater than the critical value, we would then reject the null hypothesis, okay? And we would say that there's likely then some form of gene linkage occurring or some type of inheritance that is not following our general Mendelian rules. All right, folks, good luck with that one.